Hello, everybody. My name is Andrew Strohman. I'm an MD PhD student in Wynn Lagon's lab at the Fralin Biomedical Research Institute of Virginia Tech Carilion. And today we're going to be going over some preliminary analysis of our fMRI study, looking at single element low intensity focused ultrasound to the left dorsal anterior insula and its ability to alter functional connectivity of the human salience network. The insula, located deep to the lateral fissure and covered by the fronta opercula, can be generally subdivided into an anterior and a posterior section, with individual gyri constituting each one. It receives the majority of disynaptic spinal thalamic projections posteriorly, and then integrates these bottom-up nociceptive signals with top-down cognitive and emotional pain signals, in part through communication with the hypothalamus and amygdala. On the right, you can see the attempt of a figure-of-8 TMS coil to target the insula. Notice how it lacks both the spatial selectivity or the depth penetration to target the insula. However, with free water tank data placed to scale over a structural MRI using our transducer, you can see we have single gyri resolution with low-intensity focused ultrasound, or LIFU. In a seminal study on the fMRI signature of acute pain, the authors identified both the insula and the anterior cingulate as core regions that respond to pain, as well as showing that the anterior insula and the anterior cingulate have both high sensitivity and specificity to pain versus other non-painful stimuli. An additional feature of the dorsal anterior cingulate and the dorsal anterior insula is that they constitute core nodes of the salience network, which is responsible for dynamic switching between these other pain networks and responds dynamically to pain at the network level. So, can LIFU to the dorsal anterior insula, or DAI, modulate resting state functional connectivity in regions core to fMRI-based neural pain signatures? Our first hypothesis is that LIFU can be targeted to the left DAI, and our second hypothesis is LIFU to the left DAI alters resting state salience network functional connectivity. For our protocols, we used a single element focused transducer that was stereotaxically targeted to the left DAI using the BrainSight neuronavigation system. We used co-registered MRIs and CTs for each participant for the purposes of K-wave modeling. And then we introduced white noise that was played through headphones to account for auditory confounds, and participants were queried following each session to ensure auditory masking. We had a fundamental frequency of our transducer at 500 kilohertz, and we used a duty cycle of 36%, pulse repetition frequency of 1 kilohertz, and a burst duration of 1 second. For our resting state study, we individually thresholded all people to a 6 out of 9 pain and then stereotaxically targeted to the left DAI. Participants all then underwent a 5-minute resting state fMRI followed by a counterbalanced quantitative sensory testing protocol, or QST protocol. You can see down here that for the QST protocol, we had a heat probe that could deliver transient heat stimuli to the right hand, strapped to the dorsum of the right hand, and then participants rated their pain on a scale of 0 to 9 using a 4-button box and a sliding scale that was projected onto the MRI screen. Participants then were delivered 100 stimulations with a fixed ISI of 5 seconds, and they were randomized into one of 3 arms, either an active sham to the vertex or one of 2 intensities. This was then followed up with another counterbalanced 5-minute resting state and QST probe. Our primary outcomes was functional connectivity of the left DAI with the right anterior insula, the anterior cingulate cortex, the hypothalamus, and the bilateral amygdala. Secondary outcomes were perceived pain rate. Here are the results of our K-wave modeling. This is a single participant. On the left, you can see a sagittal scan of the insula and the results of our modeling. You can see the 2 long posterior gyri followed by the anterior gyri. And you can see that with a peak pressure of 200 kilopascals, we do in fact have single gyri resolution. You can also see this on the axial scan. On the right, you can see the results of our tank data and the K-wave simulation. On the top, on the top left, you have the tank data, and then right next to it, you have the results of the K-wave modeling. Below, you have an overlay of the full width half max, showing that we have good agreement between our tank data and the simulation. In order to analyze the resting state, we combined both intensities into a single group and first performed an ROI to ROI network functional connectivity analysis with predefined regions of interest being the bilateral insula, the anterior cingulate, the bilateral amygdala, and the hypothalamus. All of these colors represent decreases in functional connectivity, with darker colors showing greater decreases. The most significant effect was decreased connectivity with the contralateral anterior insula, followed by decreased connectivity between the anterior cingulate and the hypothalamus and bilateral amygdala, followed by the left anterior insula with those same 3 nodes, and then least of which was the left anterior insula to the anterior cingulate. This is another way of looking at the same data. You can see the effect sizes from pre in blue and post in red. Just notice that the red bars are lower than the blue. And then on the right, you can see the gray bars between the left anterior insula and the anterior cingulate, showing that these did not maintain significance following our statistical correction. We followed this up with a seek to voxel analysis using the exact same left anterior insula node and identified decreased connectivity with the left precuneus. So looking back at this fMRI-based signature of physical pain, the authors also identified the precuneus as one of the core anti-correlated regions with pain. So in conclusions, single element focused 500 kilohertz LIFU confers spatial resolution to successfully target the left dorsal anterior insula. LIFU to the left DAI decreases network functional connectivity with the right AI, ACC, hypothalamus, and bilateral amygdala. LIFU to the left DAI decreases connectivity with the left precuneus. Couldn't have done this without an amazing team and a fantastic mentor. And thank you to the Focus Ultrasound Foundation and Seal Innovation Fund for supporting this work.